everyone from around the country, and welcome to today's seminar. Firstly, I'd like to take a moment to acknowledge the traditional owners and the custodianship of the lands on which we meet today. I pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australian and global society. I would also like to mention that it is National Reconciliation Week. In 2024, the National Reconciliation Week's theme is Now More Than Ever. The theme was chosen as a reminder that the fight for justice and the rights of Aboriginal and Torres Strait Islander people must continue. The week is also a time to learn about our shared histories, cultures, and achievements, so check your local university's website for events, and Reconciliation Australia, of course, also has an events calendar up on the web. A few remarks about general housekeeping. The seminar is fairly informal. We hope for your engagement and participation. There will be time for questions after the presentation. Because it's a hybrid event, we will split the question time between the live audience and the in-person audience. For everyone who's joining us on Zoom, please enter all your questions for the speaker in the chat. We will then read them out for the speaker to answer after the presentation. And everyone who's in the audience in person in the QBI auditorium, please raise your hand to ask a question. You'll be given a microphone to speak. So don't just start talking. This is to make sure that the online audience can also hear your question. So can I now please ask everyone online to mute yourselves and turn off your video? And we recommend for this seminar that you use the gallery view. The seminar will be recorded, so if you don't wish to appear on the recording, make sure your camera remains off during the entire seminar. Today, we'll be hearing from Professor Lane Strathearn, who will be talking about the impact of social experience on child development in autism. The seminar was initiated by LCC Chief Investigator Karen Falk, who is a professor and group leader in child development, education, and care at the Queensland Brain Institute. She will be moderating the discussion on the ground in the auditorium today and also tell us a few words about Lane and today's seminar. And with that, I'll hand over to you, Karen.
Thank you. And I'd also like to acknowledge the traditional owners. Today from in Queensland, at the University of Queensland, we're on the unceded lands of the Yugara and Turrbal people, and I pay my respects to elders past, present and emerging, and acknowledge the place that we are on in all of the sites today that across the 3 states have always been places of learning and research and innovation. It's great to have Lane Strathearn here today. He is an alumnus of the University of Queensland. He's a graduate of the UQ Medical School and is currently Professor and Physician Director of the Centre for Disabilities and Development at the University of Iowa. He has an endowed chair in paediatric neurodevelopment and also serves in the Division of Director for Developmental and Behavioural Paediatrics. He has a clinical practice as a developmental paediatrician and is focused on the diagnosis of treatment with autism spectrum disorder. In his training in Brisbane included a fellowship in child protection and neurodevelopment, followed by a fellowship in developmental paediatrics at the Texas Children's Hospital in Houston, Texas. Both in Australia and here, both in Australia and in the US, he's been involved in longitudinal studies, close to my heart, and in clinical science on the interface between social experiences and attachment and how they affect child development. So I'll probably stop there, but his PhD studies included the working on the longitudinal study in Queensland, the Mater University of Queensland study of pregnancy, looking at child-mother attachments using functional MRI. And he's done a whole range of research looking at things like intranasal oxytocin and its effect on brain reward systems. So he's currently, we met earlier this morning with my group, he's currently initiating a longitudinal study that's very exciting on autism and long-term effects, which is funded by the NIH, and it's entitled Epigenetics and Social Experiences in Autism, Discovering Modifiable Pathways for Intervention. So I look forward to hearing from Lane and for your engagement once he's given his talk. Thank you.
Thank you so much, Karen, for that introduction. And it's great to be here. Great to be here with you. Let's see if I can. You hear me okay? Great. Well, it's wonderful to be back in Brisbane. It's been 20 years. We left 20 years ago when our son was a newborn baby, 10 days old, when we got on the plane to go to the US, where I did a fellowship in developmental behavioral pediatrics at Texas Children's Hospital. And it's been quite a wild adventure since that time, but it's wonderful to do the full circle and come back to Brisbane and to be able to share with you some of the interesting things that I've been able to accomplish both here in Australia and in the United States over the last few years. So this is actually my grandson. And he had the opportunity of, or we had the opportunity of having him stay with us over COVID with his parents. And the one thing that just stood out to me again and again was just how incredible this process of development is over the first couple of years of life. It really is a miraculous thing. When you think that these infants are born completely dependent on us, they cannot survive without their caregivers. And yet after a very short time, they're telling us what to do, okay? They're getting into everything. They're crawling, they're walking, they're grabbing stuff, they're telling us things. And that's in the space of such a short period of time. So as a developmental pediatrician, that has really been fascinating to me to try and understand what is happening in that space, in the brain of an infant, and in that infant's surroundings that leads to this incredible unfolding of developmental capacity. So today I'm going to talk about how social experience in particular has an incredible impact on child development. So I'm going to start just by giving you a brief overview of some of the biological mechanisms that are involved in this process of child development, and then look specifically at some of the environmental factors that impact development. Attachment, both secure and insecure attachment, and trauma, parental trauma, how that impacts that relationship that develops between parent and child. And then I'm going to dive finally into a specific example of how we're utilizing some of this information on child development to explore early predictors of autism and specifically how epigenetic factors may play a role. So that's an overview of some of the territory that we're going to cover today. So first off, biological mechanisms. When you think about the fact that an infant's brain doubles in size over this first year of life, what is happening during that process? Obviously, there is an explosion of neuronal connections over that early period of time. And then there's a relative period of pruning where those connections decrease in number. So at the start of development, it's as though the infant brain is wired to take in whatever sensory information from whatever source it possibly can get, whether it's auditory, visual, tactile, smell. All of these senses are being processed in an infant's brain to make sense of that environment. But it's only those connections that are repeatedly stimulated over time that are strengthened and that are maintained. Whereas those connections that aren't being stimulated, it makes sense, why have them? So those connections are lost. And that's where there's this relative pruning process over the first few years of life. So we know that right from very early on in development, infants are programmed to seek out social cues. And that makes sense, doesn't it? Because it's from those social experiences that infants learn about their social environment. So, for example, studies have shown that when infants hear their own mother's voice, they will preferentially look toward that sound as opposed to someone else's voice that is very similar in other respects, or other sounds other than voice. The same with smell. They recognize their parent's smell and will preferentially turn to their parent's smell. With eye gaze, they will preferentially look at eye gaze that is directed toward them, as opposed to facial images where the eye gaze is diverted. And this is from very early on in development, first few weeks of life, we're seeing these differences in child preferences. You may have seen some of the experiments that have used dots, moving dots that represent biological motion. So it's these dots moving like a human being, either walking or doing star jumps. And then they just rearrange those dots so they may be upside down or randomly ordered. So they're still moving the same degree, but not in a pattern that represents human motion. Infants will preferentially gaze at biological motion as opposed to other forms of motion from a very early age. So again, this idea that infants are programmed to seek out social information in their environment. This final example of face patterns. So you can see here just a configuration of shapes that resembles a human face. On the right is a similar configuration that doesn't look like a human face. Infants will preferentially look at the face-like configuration over the other configurations. So what is it about these social cues that are so critical that we have them embedded in our developmental process and that infants will preferentially look at those cues? Well, we know from social cues is where we gain an understanding of emotion. Okay, so how do we know that a sad face represents a feeling? Well, when infants are sad, what do? They cry. Okay, that's their only means of communicating at that age. And what do we do when an infant cries? We usually pick them up, put them in our arms and go, oh, poor baby. Okay, so our face is reflecting how they are feeling. And they looking up and seeing this facial expression that's reflecting their emotion at that point in time. So it's making these connections. That may happen again and again and again over minutes, hours, days, months, gradually making that connection between our facial expressions and emotions. So all of these things are happening at a very early age in development. And of course, this development is what we call experience dependent. Okay, we use those terms in animal models, don't we? There are certain required experiences that are required for a particular pattern of development. And so it's a two-way street, isn't it? The infant provides cues, the parent responds to those cues, and then the infant responds to the parent's response. So it's sort of like a dance, okay? My wife and I used to dance tango, okay? Chest to chest, moving together on the dance floor, okay? One person is leading, the other person is following, but each person is equally important to have a beautiful dance. If one person is pushing the other person, it doesn't work. If one person isn't paying attention to the other person's cue, it's pretty bad, let me tell you, okay? So it's like that relationship between parent and child that enhances and promotes social cognitive development. So when I was in medical school a long time ago, genetics was everything. We thought that the genetic code programmed everything about human development. Okay, since that time, we've learned that development is far, far more complex than what can be encoded in the DNA. And so we've gained greater understanding of how there are other factors that intersect between the environment and gene expression. And we've called these epigenetic factors. So these epigenetic factors are influenced by social and environmental factors, such as drug use, pharmaceuticals, aging, diet, but also social experience impacts these epigenetic factors that bind to regions of the genome that affect how genes are expressed, how they function. And that makes sense. We live in an environment that affects our development. You know, if you smoke, you're more likely to get lung cancer, aren't you? If you eat fatty foods, you're more likely to get overweight. Okay, the environment affects how those genes are functioning in very real ways, and such is true for social development as well. So one example of that is DNA methylation, where a methyl group attaches to the DNA. It changes the configuration of the genome and how well that gene is able to be expressed and function. So this is how we used to see things. The genome programmed how the cell functioned, which affected how the organism behaved, which affected the environment. That's what we used to think was the sum total. Now we know it's a two-way street, that the environment is affecting the organism, which affects cellular function, which affects the genome through many of these factors such as DNA methylation, and it's back and forth. So it's opened up this whole new world of understanding of how genes interact with the environment in important and predictable ways. We've also had the opportunity to gain a great amount of understanding about social development through animal models. Obviously, in animal models, we can control the environment much better than we can with human infants, okay? And we can physically look at what's happening in the brain. How are these methylation patterns changing in the brain in response to specific environments? And many studies over decades have demonstrated that these early social experiences predict neuroendocrine development, from stress regulatory systems to oxytocin and social development. And in various animal models, including primates, rodents, and sheep. So one example of the epigenetic mechanisms that regulate social development is in the rodent model of maternal caregiving behavior, where rodents exhibit a very patterned and characteristic behavior of licking and grooming their rodent pups. So the licking and grooming has been shown to be associated with differences in DNA methylation of particular genes that regulate stress and also regulate social behavior. And that's through differences in gene expression. So one example is the glucocorticoid receptor gene in the hippocampus has differential methylation when exposed to high versus low levels of licking and grooming that affects how those genes are expressed and how we have a neuroendocrine response is regulated. So stress is regulated through these differences in glucocorticoid receptor levels in the brain, which affects differences in behavior. So this cycle of regulation of stress, but also of social behavior, is transmitted from one generation to the next, partly through these epigenetic mechanisms. So now I'd like to turn to some of the experiences in humans and how that may parallel what we've learned from animal models of attachment and social development. So I took these studies of rodent models of attachment, social behavior, and tried to understand, well, what are the key pathways involved in the brain that promote social behavior. And I found that dopamine seemed to be a key player. Dopaminergic, the mesocortical limbic dopamine pathway, which processes reward development, and also the nigrostriatal dopamine pathway, which is more involved in habit formation. So I created this model and see if we could test it in human mothers and infants using functional MRI. So we designed a study where we recruited first-time pregnant women during the 3rd trimester of pregnancy. We conducted what was called the adult attachment interview, looking for the parents' own perception of their relationships in life and how that influences their daily behavior, their social relationships in their current life. And then after their babies was born, we brought them in at around 7 months of age for a standardized interaction procedure. First, they were separated for 20 minutes, the mother and baby. Then they came together for a free play interaction session and a modified still face procedure. At each of these time points, we did a blood draw to to measure oxytocin in the blood. And then finally, there was a second period of separation between mother and baby. Then at around 10 months, we brought mums back for functional MRI scanning session, in which we showed them pictures of their own baby smiling or crying and compared to an unknown infant face. Then finally, at 14 months, we conducted an assessment of child attachment using the strange situation procedure. So this is a quick summary courtesy of ABC.
A baby's smile. It's guaranteed to melt a mother's heart. But now new research shows it also lights up her brain. Smile! Researchers at Baylor College of Medicine in Texas looked at brain scans of first-time moms as they in turn looked at photos of babies, their own and others. Some infants were smiling, some crying. At the sight of their own baby's smile, mom's brain lit up dramatically. But even the goofy grin of an unknown baby activated so-called reward centers of the mom's brain.
There really is something special about cues of babyishness. We respond with warmth to puppies and kittens, and they have some of those same cues. They have large heads, they have large eyes, they have rounded facial features, just like human infants do. This natural warmth we have toward infants is biologically deeply rooted in us.
Here's how it works. When a mom sees a picture of her smiling baby, dopamine, a neurotransmitter, is released in her brain. The areas that light up are similar to the response in a drug addict's brain. That yummy smile produces a natural high, demonstrating that we're programmed to care.
Well, what the research shows is that the smile, just by itself, just seeing it, is a powerful social reward. And it's a social reward especially for mothers who are invested in nurturing and helping that baby to feel good.
It's okay. But what about those baby tears? The researchers were surprised to find that moms did not have an increased response to pictures of babies crying. But that's probably because they couldn't hear this.
Which gets through to everyone. But some of the mothers didn't light up as much for the smile as others. What does that tell us?
I think that's what is going to be so interesting. The next step is going to be looking at these individual differences, because we know from research that there are things that interfere with that natural high, depression and stress, and supporting mothers early on is so critical, not only to their experience, but most importantly to their baby's brain and emotional development.
Okay, so that was a brief summary that highlighted some of the findings of our study, where we showed these baby faces to mothers in the scanner. Half of the faces were their own baby, half were from an unknown baby that they'd never seen before, half were smiling, half were crying. And we just looked at how the brain responded to those different facial cues. As mentioned in the video, the real question, though, was how things differ. We showed overall that mothers showed this activation of dopamine-associated reward regions of the brain, and that's what we hypothesized would be the case. But then we looked at differences in attachment based on the parent's own experience growing up, their relationships in childhood, trauma that they'd experienced, abuse, neglect, and how that may have affected how they responded to infant facial cues. When we looked specifically at happy faces, own versus unknown, we saw greater activation of mums with secure attachment in the striatum, this dopamine-associated region of the brain. We also showed greater activation in secure attachment in the medial prefrontal cortex, one of the regions that dopaminergic circuits reach out to. But what about sad faces? So when we showed sad faces, we still saw this differential response in the ventral striatum, with mothers of secure attachment showing greater activation than mothers with what we call insecure dismissing attachment. However, when we looked at this area called the anterior insula, we showed greater activation in mothers with insecure dismissing attachment. So what do we know about the anterior insula? Well, it's an area that other studies have shown is associated with feelings of pain. So not just emotional pain, but physical pain. That is an area that's often strongly activated in response to pain, or feelings of disgust or negative affect. So here we have these mums, when they see their crying baby's face, it activates these feelings of pain potentially based on other studies. And I don't know about you, but when I feel pain, I try to get away from the source of that pain. Okay, and that works when that painful stimulus could harm you. But when that painful stimulus is your own infant, that has a lot of potential implications, doesn't it? So next we looked at these levels of oxytocin. Now we all know that oxytocin has been associated with breastfeeding, with labor and delivery. Every woman who's had a child gets a dose of oxytocin injected after delivery to expel the placenta. So it has these peripheral effects associated with childbirth and lactation. But studies have also shown that there are central pathways of oxytocin in the brain that seems to be associated with affiliation, bonding, connection. So when we looked at peripheral levels of oxytocin in these mums across this period where they were a baseline separation, there was no significant difference between the two attachment groups. However, when they came together again and were interacting together, we showed that mums with secure attachment had a relative increase in their peripheral oxytocin levels compared to mums with the insecure attachment. This persisted during this mirror interaction, but by the time that they were separated again, there was no significant difference between the two groups. Okay, well, this is just an association. But when we looked at how those differences correlated with brain activation, we found that there was a significant correlation with this oxytocin region where oxytocin is produced in the hippocampus. And we also found a positive association with oxytocin response and activation of the ventral striatum, a dopamine-associated region of the brain. So the greater the oxytocin response from baseline to interaction period, the greater activation we saw in these two crucial regions of the brain when mums were seeing pictures of their own baby. Next, we looked from the adult attachment interview to measures of unresolved trauma. So unresolved trauma is defined as where there has been a traumatic experience in someone's life, whether it be child abuse, neglect, sexual abuse, abandonment, but not just the experience having occurred, but it having an ongoing influence on that person's daily life in the present. So that was our definition of unresolved trauma based on coding of the adult attachment interview. So we were particularly interested in an area of the brain called the amygdala, which is often involved in the processing of emotions, particularly negative emotions. So we simply compared mothers who had no history of unresolved trauma and those with unresolved trauma currently. And we looked at activation of these amygdala regions in the brain for these different infant stimuli. So own infant, unknown infant, happy and sad faces. So first, when we looked at those without any trauma, we found a relatively expected response. There was slightly greater activation for sad affect than for happy affect in the amygdala, and that's what other studies have shown. And this is looking at an unknown baby, so any baby face, just showing sad faces and happy faces. When we looked at their own baby's face, it was a similar pattern, but much more exaggerated, so a much stronger amygdala response for their own baby's sad face compared to their own baby's happy face. And overall, that response was greater than for unknown baby faces. Then when we looked at the group of mothers with unresolved trauma, when they were shown pictures of unknown baby faces, a similar sort of pattern, but generally elevated. So greater response to sad faces than happy faces, when they were shown unknown baby faces. But look at the difference when we showed them their own baby. So there was a strong response for their own baby's smiling face of the amygdala, similar sort of pattern for those without trauma. But look at what we saw when they were shown pictures of their own baby in distress. It was almost as though the amygdala was being shut off, okay, that they were turning off that response when they were seeing their own baby's distressed face. Now remember, these are women who had experienced trauma in their own childhood that was unresolved at the present time, and now they were seeing their own baby in distress. It wasn't true for any baby in distress. It was specific for seeing their own baby's face in distress. So then we looked at a group of high-risk mothers. So these are women who have used substances, who have had substance addiction during pregnancy. And we compared their response to seeing their own versus unknown happy baby face in two areas associated with dopamine circuits, the ventral striatum and the ventromedial prefrontal cortex. Between 0 and 2 is the time window where the faces were being displayed on the screen. And the green graph is their own baby's happy face. The red graph is an unknown baby's happy face. So as expected, we saw this increase in response when they saw their own baby's happy face that wasn't present for the own baby's sad face. Look now at what we saw with mothers with addiction problems. Okay, so you'll notice there's an inversion. It's like it's upside down, where they're not showing a greater response for their own infant's smiling face, but more so for this other unknown infant's face. When we looked at the attachment interviews of these mothers that we'd recruited with addiction problems, around 98% of these women had unresolved trauma. But almost all of them had this pattern of unresolved trauma. So it seems as though instead of getting a reward response when you see your baby, they're certainly not getting that. And from that other study of the insula, it seems as though they may be withdrawing from their baby when they see a sad infant face. So this has a lot of important implications for me as a developmental pediatrician. So when I was in Brisbane decades ago, I did a fellowship in child abuse and neglect. And so we often saw parents who were neglectful of their parents. And that's a very stigmatizing thing, to label a parent as being neglectful of their own infant. However, this data has helped me to gain a better understanding of what might be going on underneath neglect. If these parents have experienced trauma themselves and they're not processing their infant cues the way most people do, in fact, they're getting a pain response when they see their infant's distress, or they're not getting that motivating reward response to continue to engage with their infant, then that may help us to better understand why, in some cases, it results in child neglect. So I talked a little bit about epigenetics, how it may be a mechanism that helps us to understand the connection between social environment, social experience, and what's happening under the skin. So I'd like to tell you a little bit about a project that we've just been funded to conduct. So we're just in the process of starting this research in the US, looking at epigenetic markers and how social experience may change epigenetic factors and have an impact on social development in autism. But first off, let me just remind you of the numbers, what we're seeing all around us. And I know these data points are from the US, but I know a similar pattern is being observed in Australia, where more and more children are being diagnosed with autism spectrum disorder. So over the last 20 years, there's been this massive rise in the incidence of autism. And of course, a lot of that is because we're more aware of the challenges of autism, that diagnosticians are more apt to diagnose, to recognize and diagnose autism in children and young adults. This paper looked at some of these factors that may help to explain this increased prevalence of autism. We're seeing higher parental age, which may be associated with autism. But still, there's almost half of the cases where we just don't have a good understanding of why the prevalence is increasing. And so that's led us to develop a model in which we're looking at this social spectrum of experience between pregnancy, birth, and an autism diagnosis. Now, we know there are intrinsic factors. There's been many, many studies looking at genetic markers that are associated with autism. And so there's this direct connection with autism. But we're also thinking, well, what's happening with DNA methylation across time? How are these patterns changing? And how does genetic variation influence methylation patterns? Because other studies have shown that genetic variation itself can affect patterns of methylation. But then the other part of this equation is what are the social or extrinsic factors, such as social experience, things like prematurity, family environment, daycare settings, social experience outside of the home. All of these things may be influencing social development through these epigenetic mechanisms. So this is what we're planning to test in our new study. So we have been funded to recruit over 7,000 pregnant women before the 3rd trimester of pregnancy. And we'll follow these mothers and their children for the first 2 to 3 years of life. We've developed a smartphone app called Baby Steps that helps us easily collect data over the smartphone. So questionnaires, ecological momentary assessments to see how things are from moment to moment, from day to day, week to week. After the babies are born, we're asking these parents to do what every parent already does, and that's videotape their baby, so that we've got this longitudinal, ongoing record of infant social development. And then from the genetic or the epigenetic point, we're looking at epigenetic change from birth to 18 to 27 months, and then a 3rd blood sample will be taken at 6 months later. So we're accessing the child's newborn blood samples to look at epigenetic markers at birth compared to around the time of diagnosis. And then we'll be looking at 3 specific groups: those who are diagnosed with autism spectrum disorder, children who have developmental delays without autism, and then a group of typically developing children. We're particularly interested in oxytocin for the reasons I've discussed. So we'll be doing a deep dive on the oxytocin receptor gene, looking at pyrosequencing to looking at methylation across the oxytocin receptor gene. We'll be doing epigenetic clock analysis, which is a marker of developmental age across time. And then finally, looking at RNA expression at these DNA methylation sites, just to get a clearer picture on what's actually happening at the level of gene function when these genes are increased or have increased or decreased methylation. And then looking at polygenic scores for autism. I think we're getting more and more understanding that autism is not just a single, well, we know it's not a single gene condition. There are multiple genes, and every week there are new genetic factors being published that are associated with autism. So the idea is that maybe it's multiple genetic markers that together increase likelihood for autism. So we'll look at polygenic scores. And then finally, between 18 months and 3 years, we'll do an in-person developmental assessment for autism and other developmental concerns. We'll use a recording device that picks up language experience at the home. The child will just wear this recording device in a pocket. And then there's an automated algorithm that looks at the number of times there's interactive language communication between adult and child in their environment. So our overall model is that perinatal experience, which includes prenatal stress and postnatal social experience, is a predictor of social communication deficits between 18 and 30 months of age. That there are polygenic factors that may modify that relationship, and that there are DNA methylation changes that help explain these associations both at birth using the newborn blood spots and at 18 to 30 months using the blood samples collected around the time of diagnosis. So hopefully we'll have some interesting results in the years to come, but that gives you a little bit of an overview of what we're planning to do at the moment. Whoops. So, any questions?
Thank you very much, Lane, for that fascinating talk and very accessible. That was great. The online chat here has so far been quiet. Karen, do you see raised hands in the auditorium?
Yes.
Then I'll maybe start with the in-person questions. There are now online questions coming in.
Thanks very much. Thanks, Lane. That was a fantastic talk. I'm glad I've got my child rearing out of the way. And it gets back to my question, which is about stress. You know, we live in an increasingly stressful world, and there are increasingly people that are opting out of having children or having anyway. Here it's back again. Yeah. So, does your study take into, or any other studies that you've either authored or been part of, taken into account just generalised stress as a causative of the rise of, you know, ASD and other neurodivergent kind of behaviours, and in particular, and I'm not trying to be nasty here, but in the US. In states where women are actually very stressed when they're pregnant, you know the reasons. I don't even need to say them. Are these factors being taken into account in these studies?
Yeah, well, that's definitely one area that we're focusing on, is prenatal stress, but how that carries over into the postnatal environment as well. From other studies, I know there's been a lot of animal models looking at prenatal stress and its effect on neurodevelopment in the offspring. So there's a lot of interesting data that supports that hypothesis that stress during pregnancy actually affects neurodevelopment, developmental pathways, neural pathways in the brain of the offspring. And of course, prenatal stress is a pretty good predictor of stress postnatally as well. And so we'll be trying to get snapshots of that. Of course, this is parent report. Other studies have done more biological measures of stress to look at and tie that in with developmental outcomes. Because we've got such a huge sample that we need in order to come into that subgroup of children with autism further down the track, we're trying to keep it as simple as possible during that first phase. So it's just app and videotape responses. But I think we'll get a pretty good measure of stress from those measures, the parent report and also the video assessments.
When you were talking about the increased prevalence of identification of those children with ASD.
Would you please use a microphone for your question? The online audience can't hear. Thank you.
I can repeat the question because it's, well.
Thanks.
We're doing a switch in microphone, so hopefully this will be better.
Better. Increased prevalence of identification of those with ASD. From my understanding, for a long time, it was very difficult to diagnose females with ASD because they were so good at masking. Is that one potential one? And is that a consideration for your analysis in comparisons in your study? And further to that, with ASD in Australia, there's levels 1, 2, and 3 with this stratification. Will you be looking at those epigenetic changes in those sort of levels to see if they correlate?
Yes, yes. So firstly, the male-female distinction. I think that's a really important area that we're gaining more understanding of now, but it's still fairly rudimentary, understanding these differences between how males and females present with autism. The problem with autism is that it's us who are defining what is autism. Okay, there's no biological measure that says this person has autism, this person doesn't. And those definitions have evolved over time, have been more encompassing, and that's part of the reason why we may be seeing more cases of autism. But it's the same with the distinction between males and females. It's what do we define as autism? What do we include within that umbrella? And that's really just based on our clinical experience and not on biology, unfortunately. So it's a really important but difficult to tackle question.
We've got some online, one more question in the audience. So Barbara, do you want to go to the online?
Yes, thank you. So we have two quite connected questions. One was asking what happens when you help parents resolve their trauma for the next generation. And the other one, whether you looked at whether provision of pain relief changes activation levels in mothers with insecure attachment.
I think I may not have got it exactly right, but with the unresolved trauma, the question was about the next generation, what some of the consequences may be. Is that right?
Yeah, so I think it's asking about, can we basically help parents resolve their trauma to not continue the cycle?
Right. So a very, very practical question. So one of the classifications of in the adult attachment interview, the model that I use is resolving, reorganizing attachment. So that means that they still have evidence from the interview of insecure attachment, but they're moving toward security from that interview. So we did a study and just looked at how mothers with reorganizing attachment, meaning heading towards security, how that was associated with security in their offspring's attachment. And we found that there was a stronger association with secure attachment in those mothers with reorganizing attachment. So that's a hopeful thing, that if we can help people to understand the impact of trauma on their current level of functioning, to gain a perception of that and how those experiences may have an impact on their relationship with others, so that they can reorganize their perception, then that may have positive implications for their offspring. So that was just one example of a study that we've published. The second question was about
pain relief, if that can help.
If what, sorry?
If pain relief can help.
Pain relief. Oh, if that can help.
With
Okay.
With the pain response.
Okay. Well, this is an interesting question as well. And another study that I was involved in looked at women who had used pain relieving medication postpartum after a C-section. Okay, so routinely they're prescribed strong pain relief medication after a C-section, obviously, and they used prescription records, publicly available prescription records, and they showed that those women who went on to require longer pain relieving medication, so more prescriptions over a longer period of time, and we know that there is a problem with addiction from starting out with simple pain relieving medications. The factor that was most strongly correlated with ongoing use of pain relieving medications after C-section was depression. So nothing to do with the physical pain, but pre-existing mental health concerns. So I think that may help us to understand this association between activation of this pain region, the anterior insula, and differences in response to negative affect from the mother's babies. We know that this region of the brain, the anterior insula, responds not just to physical pain, but also to emotional pain. So if you've got the same area of the brain that's processing pain, whether it's physical or emotional, if you have a physical need or a physical pain, like a C-section recovery, you're given medications that ease that pain. The wound heals, but if there's still emotional pain that's not going away, then perhaps that would explain why some people are more likely to continue to use these pain medications. So going back to the original question is, does pain medication, could pain medication help? I don't know. But I don't think the medication is addressing the real source of the pain. Okay, it's not treating the source of the pain when that pain is related to trauma or abuse.
Yeah, that does make a lot of sense. Can we maybe have one more online and one in the auditorium, and then let's go online.
One in the auditorium.
Pardon me?
She said, go ahead with the one online and then we'll finish with the one in the auditorium.
That sounds great. There's another question from Shannon. How are you thinking about in measuring parent-child interaction during sleep at night and napping in your longitudinal study?
Parent-child, well, we're just measuring parent-child interaction during the day. So we're asking them to have a free play session with their child lasting, I think, 5 minutes, and they'll videotape that on their own phone, another parent, or just setting up the phone. And then that pattern of interaction will be coded over time. So I'm not sure about the relationship with sleep there.
I don't know if any of our sleep researchers want to come in.
No? Okay. Maybe just last one and quite quickly. Going back to the trauma, was there a weighting or a scaling of the level of trauma and the effects seen in the insula in regards to the happy or the sad known baby phase? Like more trauma equals more deleterious effects compared to less?
Yeah, unfortunately, we didn't look at that level of granularity. We just had a dichotomous yes or no to unresolved trauma, but that's a really interesting question. Maybe sort of related to that is when we were looking at the addiction group, mothers who'd used substances in pregnancy, we did not find a relationship between their level of addiction and their level of dopamine reward activation. So remember, we saw that the addicted mother didn't show the normal increase in reward activation. You would expect if it's an addiction, if it's truly related to addiction, then the more addicted they are, the more substances they're using, the worse their dopamine activation would be. We did not find any relationship whatsoever between level of addiction and brain response to reward. And that makes me think that maybe the addiction is just one of the byproducts of early life trauma. They're more prone to use substances, but that's not what's driving the problem, the interpersonal problems with them and their children.
So Barbara, I think we're finished now, but I'd like to say thank you on behalf of everyone here in the audience, and thank you very much, Lane. Hope we can get you back next year. And I know you come regularly to Brisbane as you have family here, so that's a nice opportunity to hear how the study is going. I'll hand over to you, Barbara, to finish.
I can just second that. Thank you very much, Lane, for a fascinating talk and for answering all those questions. There are a few more that we got in the chat. If I can forward them to you, and maybe you can get back to some of the participants, that would be great. And yeah, thanks to Karen for initiating it, and thanks everyone for joining us and participating in the discussion. Have a great day.